Hello, everyone. I hope you can hear me. So it's my very pleasure to start with today's geneXplain webinar. And I'm particularly happy that we really got an exciting talk for today. And I'm very glad that Dr. Tal Iram has accepted our invitation to show us her latest results on young cerebrospinal fluid restores oligodendrogenesis and memory in aged mice via Fgf17. The results have been presented recently in a paper published by Nature and made it even to the general press and broadcasting and TV services and raised a lot of comments, I think, worldwide. Dr. Tal Iram is the postdoctorate fellow at the Department of Neurology and Neurological Sciences at Stanford University School of Medicine in California. She received her PhD in neuroscience from Tel Aviv University in Israel in 2017. Shortly after, she began her postdoc in the Wyss-Coray lab at Stanford, where she developed a novel rejuvenation paradigm by transferring cerebrospinal fluid from young to aged mice. This is the essence of her talk, but there will be way more to learn. And we are all very excited and looking forward to your presentation. Tal, please start.
So hi, I would like to start by thanking Edgar and the team for the great opportunity to present our work here. I'll start sharing my presentation. Yeah, so as Edgar mentioned, I'll describe our recent study focusing on young cerebrospinal fluid and showing that by transfers of cerebrospinal fluid from young to aged mice, we can affect a cell type in the brain called oligodendrocytes, which I'll deep dive into in a moment. So in the first part of the presentation, I'll lead you through the main findings of this paper. And in the second part, I'd like to share unpublished data from a study that stemmed out of this initial observation, where we focused on the role of this transcription factor, serum response factor, in the basic cell biology of oligodendrocytes in the developing brain. So just to set the stage, I'd like to mention that aging is the primary risk factor for major human pathologies, including cancer, diabetes, cardiovascular disorders, and neurodegenerative diseases. And with increasing lifespan in the general population, these diseases pose an immense societal burden. This graph here shows that the increase in the comorbidities between these diseases with age in the general population. And you can see that there is the period with no diseases, which we term in the field health span, decreases dramatically with age. Interestingly, there's a very small proportion of the population that remains resilient to all of these diseases. And we're very interested in these individuals to study their resilience mechanisms to hopefully apply them to the rest of the population. I think that this graph also shows that targeting aging as the upstream cause for disease could potentially shift this all graph to the right, thereby delaying the onset of disease and dysfunction. And this notion has led to extensive research and development of various strategies in model animals to target aging and to delay its onset, and in some cases to seemingly reverse it. One such rejuvenation study is achieved by the surgical connection of a young and an aged mouse in a surgery termed heterochronic parabiosis, where these young and aged mice will share the blood circulation. Another strategy could be achieved by transferring of plasma from young to aged mice and vice versa. And these Repeated plasma transfers were done by the Wyss-Coray lab and many others in the field, and they demonstrated that blood-borne proteins hold the potential to reverse various aging hallmarks in many tissues in the aged mouse body, including the brain. However, the brain is unique because it's partially secluded from the blood by the brain barriers, which makes it very challenging to identify how these youthful blood proteins interact with the brain. Instead, the brain is bathed in cerebrospinal fluid, or CSF, which flows in the brain ventricles and around the brain, surrounds the brain and the spinal cord. The CSF is produced by a secretory tissue which lies within the brain ventricles. It's called the choroid plexus, and it provides mechanical cushioning to the brain, and is also composed of ions and proteins that match the brain unique needs. So this made us wonder, could young CSF be used to rejuvenate aged mice? And if so, would it simplify our understanding of how these proteins interact with the cells in the aged brain? So to perform that in a functional way, we took a step across those brain barriers directly to the brain and collected CSF from young mice and slowly infused it directly to the lateral ventricles of aged mice using these mini osmotic pumps. These CSF transfers were never done before, mostly because of the complexity of drawing enough CSF from young mice without any blood contamination. After we set out the system, we screened these aged mice at various levels to see or to identify what are the cell types that are responding to these young CSF proteins and which are the cellular pathways that are induced by it. So at the behavioral level, we asked if CSF infusions could improve long-term memory in a paradigm of remote fear conditioning recall. In this paradigm, which is an hippocampal dependent memory task, a mouse is put in a chamber where it receives 3 food shocks in a certain context. And this is paired also with a flashing light and a tone. These mice are then infused with either young CSF or artificial CSF, which serves as control in these experiments. And 3 weeks after the initial shock, the mice are put in the same context where they received the shock, but with no shock, where we assess their memory by their level of freezing, which is the fear response in mice. And as you can see here in the graph that measures the percentage of freezing, we find that mice that received young CSF retain the memory, the fearful memory, better than controls. So this was very intriguing to us, and to gain better mechanistic understanding, we isolated the hippocampus, this region that is known to be involved in learning and memory, but also in memory-related cognitive decline or aging-related cognitive decline. We isolated these hippocampi and performed transcriptomic bulk RNA-seq of their mRNA to find what are the underlying mechanisms. And to our surprise, many of the differentially expressed genes between the young CSF receiving mice and the artificial CSF receiving mice were associated to oligodendrocytes. And this came as a big surprise to us. We were expecting to see neuronal genes or microglial genes, but it was very starking, the signal that came from oligodendrocytes. And just as a small example, I'll show you the genes or some of the genes that were changed. You can see in this graph that most of the genes were upregulating upon exposure to young CSF. And they consisted of major regulators, known regulators of the oligodendrocyte lineage cell maturation, such as olig-1 and MIRF, as well as the protein components of myelin, which are MBP and MAG, for example. This was very intriguing to us because there's evidence from human MRI studies measuring the volume and the integrity of white matter, as seen here, that white matter content increases in humans in the first 5 decades of life. And this is thought to be linked to the increasing experiences and learning and memory throughout life. But unfortunately, around the age of 60, white matter starts to break down quite dramatically. And this occurs also in Alzheimer's disease, for example, and is thought to or be correlated very tightly with the loss of cognitive function. Yet the molecular mechanisms that underlie this breakdown of myelin are mostly unknown. We know that the white matter that was measured in these MRI studies is produced by mature oligodendrocytes. It's these cells in blue that wrap their membranes around the axon, the cable part of the neuron. And by doing that, they insulate it very similarly to how an electrical cable is insulated with the plastic wrap, and thereby enabling better nerve conduction. But they also actively support this axon metabolically and by many other ways to fine-tune the neuronal network synchronization. These mature oligodendrocytes are formed in the brain in a process termed oligodendrogenesis, where an oligodendrocyte progenitor cell, or an OPC, given the right cues, it will proliferate and differentiate and mature to this myelinating oligodendrocyte. And interestingly, this process was recently shown to be necessary for the formation of long-term memory and its consolidation and recall. And this was done in young mice, yet we don't know how oligodendrocyte lineage cells age and whether they causally contribute to aging-related cognitive decline. So we took a brain, this is a mouse hippocampus of a young and an aged mouse. Aged mouse is around 1.5 years or 2 for a mouse. And we stained for these oligodendrocyte progenitor cells, seen here as green dots. And one can appreciate that these OPCs, these are oligodendrocyte progenitor cells, they tile the brain, they're spread all over the brain. They don't reside in very specific niches. And also they remain in very vast numbers in the aged brain. And this is opposed to neuroprogenitor cells, for example, which are mostly depleted at these ages. Yet when we give the mice EDU or BRDU, which mark proliferating cells, we found that while the cells are still there, their rate of proliferation declines dramatically in the aged brain. So we hypothesized that there might be a factor in young CSF that is boosting their proliferation and maturation in the aged hippocampus. So to probe that directly, we infused the aged mice with young CSF or artificial CSF and pulsed the mice with EDU or BRDU to label proliferating cells, and indeed found that following exposure to young CSF, the rate of proliferation of hippocampal OPCs increased. To test if these cells actually mature to form more myelin in the hippocampus, we infused another cohort with CSF, but this time let these cells to mature for 3 weeks. And only then, which is the time that we know that takes for the full oligodendrogenesis to occur. And we then stained the hippocampi with myelin basic protein, which is a major component of the myelin. And indeed, we found that in this subregion of the hippocampus called the molecular layer, there was a stronger intensity of this stain, which indicated that there's more myelin in this region of the hippocampus. We further validated these findings by doing transmission electron microscopy, which enables to see the ultrastructure of myelin around axons. It's seen here as these black rings around the axons in a cross section. And we quantified how many of these myelinated axons there are exactly in the same region, the molecular layer of the hippocampus. And indeed, we found that there were more myelinated axons, and this supported our hypothesis that there is de novo myelination. There is formation of more myelin or myelinated axons in the hippocampus following exposure to young CSF. However, in the brain, when we do these infusions, we can never tell if this is a direct effect of young CSF proteins on oligodendrocytes. It could also be mediated through another cell. So to see if this is a direct effect, we turned to oligodendrocyte cultures, which were produced in a great collaboration with the Zucker lab at Stanford, which provided us with pure OPC cultures. And we basically repeated the same experiments that we did in vivo. We supplemented the media of these OPCs with 10% of young CSF or artificial CSF, and found very similarly that young CSF increased the proliferation of these cells in a dose-dependent manner. And when the cells were allowed to differentiate, we found excitingly that young CSF boosted their maturation and also increased the overall cell viability in the culture. And I would like to show you how these cells look like throughout the differentiation process in culture, which occurs in vitro in the course of roughly 5 days. You can see that this is the artificial CSF. You can see the cells that the OPCs are initially bipolar, and as they differentiate, they start spreading their membrane, and in the lack of an axon, they basically myelinate the cell culture dish, and they become very flat, as you can see here. And this is what we initially saw when we first added young CSF to the culture. There's a boost of proliferation and very quick maturation of the cells. You can see that they're very happy with CSF, and overall they thrive under these conditions, which is just a supplementation of 10% of their full media. So how does young CSF promote myelination? We could then use this in vitro system of the OPC cultures to try to tease out what are the very initial cellular responses to young CSF. And for that, we chose a new RNA sequencing technique. This is a bulk RNA sequencing technique termed SLAM-seq, which also enables to label newly transcribed mRNAs by supplementation of a uridine analog to the culture, which then gets incorporated in each newly transcribed mRNA. And this was the data analysis was done in a great collaboration with Fabian Kern from Andreas Keller's lab in Saarland University in Germany. So we added CSF to the OPC cultures and performed SLAM-seq after 1 hour and 6 hours to see what are the very initial cellular responses to young CSF. And the top transcribed gene seen here in the volcano plot was a transcription factor and an early response gene called serum response factor, or SRF. And I would just like to mention that at this point, we used the Transfac database to learn what are the predicted SRF target genes, and this was very helpful to interpret the data in this experiment and all of our follow-up experiments. So SRF was extensively studied in muscle cells, but also in the brain in neurons, where it was shown to mediate the migration of neurons throughout development, the formation of the hippocampal cytoarchitecture, and very interestingly, in the adult mice, SRF in neurons was shown to mediate the structural changes that are necessary for neuronal plasticity in learning and memory. Yet we don't know what is the role of SRF in oligodendrocyte lineage cells. At the cellular level, we know from other studies that various ligands or proteins could activate SRF downstream through several intracellular pathways. I would like to mention one which involves the actin cytoskeleton. And indeed, at the 6-hour time point, while SRF was back to baseline, many of its known target genes that are linked to the actin cytoskeleton were upregulated in these cells. So we could actually track the activation of this pathway. To test it more functionally, we stained for phalloidin, which stains actin filaments, both in the OPCs in culture that were exposed to 6 hours of CSF, and the OPCs in the aged hippocampus after infusion of young CSF. And in both cases, we found that there is an increase in the actin content of these cells, indicating that the activation of SRF has a functional impact on this pathway. Lastly, we knocked out SRF in these cells in vitro and found that that was sufficient to block the boost of proliferation that we found with young CSF, indicating that this effect is mediated by SRF. So this was all done in vitro in cultures from young mice, and we wanted to link this observation back to the aged brain. And for that, I would like to take a small detour to describe a collaborative project that I was involved in at that time. This was part of the Tabula Muris Cenis consortium, which was led by Tony Wyss-Coray, my advisor, and Steve Quake at Stanford, which also serves as the CEO of the Chan Zuckerberg BioHub. And they came up with this idea to generate a single cell RNA sequencing of as many organs as possible in the with aging in mice in both sexes and with rejuvenation such as parabiosis. And this has already led to various very interesting observations. I'm adding here the links to the browsers if you're interested to check genes that you're studying. And again, this was a collaboration with many labs and with various expertise in isolating single cells from various organs. And I was in charge of isolating cells from the brain. So this generated this very extensive t-SNE plot where each dot here is a single cell in a different organ. And when we overlay SRF expression, we indeed found that SRF is expressed by various tissues. And interestingly, when we measured its expression both at the expression level and the fraction of cells expressing SRF, we found that there is a decline in SRF expression in various tissues, including the brain. And this was in line with reports that when SRF was knocked out or downregulated in human or in mouse or even C. elegans, specifically in muscle cells, this led to premature aging phenotypes. So we found it very interesting and zoomed back to our oligodendrocyte clusters, the OPCs and the mature oligodendrocytes, and indeed here found that SRF was downregulated. We further validated this specifically in the hippocampus by performing RNA scope in OPCs and validated that indeed SRF levels were downregulated. So is the SRF pathway also induced in the hippocampal OPCs in vivo? For that, we basically repeated the SLAM-seq experiment, but in vivo, where we injected acutely CSF to aged mice and sorted out the OPCs and performed bulk RNA-seq of hippocampal OPCs after an hour and 6 hours. But first, we wanted to test if if the SRF pathway is affected in aged hippocampal OPCs with aging. So we isolated OPCs from young and aged mouse and plotted the overall expression of aged versus young OPCs, focusing on the predicted SRF target list by trans fat. And indeed, we find that the pathway is overall inhibited with aging. Yet after injection of young CSF for an hour and for 6 hours, many of the genes were upregulated. So we think that this activation is also occurring in vivo. So just as an interim summary, I've shown you that this process of maturation of oligodendrocytes, which is termed oligodendrogenesis, declines with age, and that exposure of the mice to young CSF increases oligodendrogenesis in an SRF-dependent manner. And this is correlated with an improvement in long-term memory recall. So next, we set out to find what is the factor in CSF that is inducing these effects. And to narrow down the list of tens of thousands of proteins that we know that are present in CSF, we noticed in the literature that many of the upstream activators of SRF are also transcriptionally controlled by it. So we used that to narrow down the list and crossed the predicted SRF targets by trans fat with CSF proteins that were identified in human CSF proteomic studies and generate a short list of 35 potential proteins. We then moved to an in vitro system with a reporter for SRF. So if the SRF pathway is activated, the cell will turn green, and added these recombinant proteins in a dose-dependent manner, one by one. One of our most potent inducers was a fibroblast growth factor termed Fgf17, which very nicely increased the activation of the SRF pathway in a dose-dependent manner. SRF is a brain-enriched protein that is necessary for the formation of the hindbrain during brain development, but not much is known about its production in the adult brain and how it is regulated with aging. So we set out to find which are the cells that are expressing Fgf17 in the adult brain by performing RNA-scope and also stainings for Fgf17, seen here as the red dots inside the green cells, which are the neurons. And we indeed found very extensive expression in the cortex, in the hippocampus of young mice. However, with aging, there was a very dramatic decline in Fgf17, both at the transcript and at the protein level. In the CSF, we mined a human CSF proteome dataset from the Montin lab at Stanford, where we found that Fgf17 levels decrease in human CSF with aging in a healthy population. So is Fgf17 sufficient to mimic the effects of young CSF? For that, we basically repeated the same experiments that we did with young CSF, but infusing Fgf17. And indeed, we found that after a week of infusion of the recombinant mouse Fgf17, there was a boost of proliferation of OPCs. And most importantly, we found that it mimicked the beneficial effects of long-term memory recall, very similarly to what we saw with young CSF. Lastly, when we fused Fgf17 blocking antibodies, so preferring the opposite experiment, we took a young mouse and blocked the function of Fgf17 by infusing inhibitory antibodies. And we found that that was sufficient to impair the memory performance of these mice, both in the contextual fear conditioning and in the Y-maze alternate assay, which are both hippocampal dependent memory tests. So just to summarize, overall, these results inform the following model where OPC proliferation and differentiation decrease with age, and re-exposure of the age brain to young CSF and Fgf17 improve or boost oligodendrogenesis concomitant with improvement in long-term memory recall. And to conclude, this scheme was generated by Maria Lehtinen, a leader in the CSF field at Harvard, which wrote news and views on our study. And I think that we're all really used to think about CSF as a proxy to the brain for biomarkers in disease, for example. But this study, as well as others from Maria Lehtinen's lab, show that there is another axis from the CSF inwards to the brain. And we believe that CSF has a functional impact on the function of the cells in the brain, and its changing composition might result in cognitive decline. And this presents a new direction for research by targeting directly CSF, potentially also targeting the choroid plexus, which produces and secretes CSF proteins, as a therapeutic approach for treatment of dementia, for example. It also highlights the importance to study oligodendrocytes in the aging and the neurodegenerative diseases. I find it really encouraging that these cells, even at a very late age, could be manipulated and their function could be partially restored. And this is in line with various other studies that showed that oligodendrocyte function could be restored in the context of aging, but also in many other diseases. And these various genetic and environmental and pharmacologic strategies could be now leveraged as a therapeutic to target directly oligodendrocytes in these diseases. And I think there's still a lot of unknowns and opportunities for further discoveries in this field. And with that, I would like to just wrap up and acknowledge Tony for being an amazing mentor, to all the lab members that contributed to this work, especially Chint Kaur, Saket Maineni, Heather Shin, and Alison Morningstar. To our great collaboration with Andreas Keller's lab and Fabian Kern at Saarland University, Brad Zucker's lab at Stanford, and Miguel Garcia, and Henrik Zetterberg for providing us with young human CSF samples, and to all of our funding sources, and to you for listening. And I would love to take any questions and feedback.
Well, yeah. Thank you. Thank you very much, Tal, for this really great talk and sharing all these very straightforward evidences with us. Just wonderful. Thank you very much. So with that, I will forward the word to Alexander, who will moderate the discussion now.
Hello. Hello again.
Hi.
So hello, Tal, and that's really a great talk. Actually, indeed, that's very important questions, especially with aging. You ask yourself how to renovate or restore it. And I was really listening it through. And let me moderate a little bit. So please, I'm asking attendees. So please put your questions in Q&A, and then we'll be ready to answer them. So don't hesitate. So we'll wait for questions. There are none so far. Actually, in between, I have a number of questions.
Yeah,
so,
sure.
Yes, of course. First question concerning, generally, of course, I'm not from that field, but I'm a bioinformatician. But we hear a lot about what happens with our brain and with aging. And the general concept was that our neurons are dying. And what you are saying, actually, that they're rather getting covered less. And the question is whether this covering is actually, which is done by oligodendrocytes and this myelination, is protecting neurons from dying, or it's something else happening there?
Yeah, I think in aging, it wasn't studied enough to be able to really answer this question. And that's something that I'm very much interested in following up. We know from demyelinating diseases, from like multiple sclerosis, for example, where it's a very different like mechanism where the immune system attacks myelin, kills the neurons and this wrapping, kills the oligodendrocytes and the wrapping of the neurons, and that subsequently will lead to axonal degeneration and dying of neurons. But it's a multi-step process, and in many initial stages, it's still reversible. And we know from multiple sclerosis that there is a relapsing, remitting phenotype where the immune system attacks the myelin, but then oligodendrocytes are able to reproduce myelin, and new OPCs mature and make more myelin instead of the damaged oligodendrocytes. So there's a lot of opportunity for repair. And I think that that's also the case in the aging brain, where at initial stages we can intervene and repair, and eventually like save these axons from being degenerated. Once the neuron is dying, there is not much we can do.
So still, you're saying it's more the final effect of the loss of the memory and all this degeneration which is happening with aging is basically because of neurons dying, or maybe just by speculating, maybe the covering itself, these myelinations, protects whatever neurons and the signaling from losing it, and itself, this protection makes the memory better, or how to test it?
Yeah, so I think memory, it's a really complex process, memory, because it requires a synchronized activation of neurons from many regions in the brain. And you can imagine that myelin, we know that it regulates the conduction speed of these neuronal signals. So by changing its width or length, it affects how well the signal is synchronized across these different regions when you're trying to recall a memory. So there is an additional layer of regulation at the myelin stage.
Yeah, I see. Okay, so we got one first question. Let me read it. So when you upregulate SRF or FGF, do you see development of any diverse side effects in the aging mice, such as tumor development, abnormal growth, or something like that? Okay, that's very
good. It's a very, very important question. And this is always the tension between when you are trying to induce proliferation of stem cells, there's always the risk of inducing tumors. I think that the advantage in this sense in the aged brain, that it's so inhibitory for proliferation, we don't see any tumor formation or adverse effect. I haven't tested in any other organs in the body, because we're administering it directly to the central nervous system, to the CSF, but potentially it's trained out to the plasma, and I think it could have effects on other organs, and that should be tested.
All right, okay. Yeah, thanks for your presentation, people sending, so that's really great. Okay, waiting for more questions in between. Talking about SRF, actually, on your first slide, which you showed the result of gene expression analysis, you got this SRF, the most important and most highly upregulated transcription factor, but there was another one also shown on your picture, EGFR.
EGR1.
Yeah. Do you know anything about it, or its role? Did you check it?
Yeah. So EGR1 is controlled by SRF. Actually, it's one of the most known target genes. But on its own, it's a transcription factor and an immediate early gene. And there's a lot of known functions that are associated to it. In our context, and also in oligodendrocyte lineage cells, so EGR1 maintains the proliferative state of OPCs, and then it's downregulated with differentiation. When I was analyzing it, because it's downstream to SRF, I wanted to focus on the most upstream regulator that I could. So we didn't follow up with any targeted EGR1 studies.
Yeah, but also it's coming to bit to my interest of transcription factor cooperation. So, and perhaps when you ask a question whether this specific expression and specific action of SRF in oligodendrocytes may be accompanied by some other transcription factors. Maybe that's something you already studied, or it's something you're going to check.
Even for SRF, it's very complicated because it has known cofactors that lead it to different target genes based on which upstream cellular pathway is activated. I highlighted only the actin genes, but there are others, and there's other cofactors that are involved there that will bind to SRF, and that will determine which target genes are being activated. So even at the SRF level, we know that these cofactors are expressed in a cell type specific manner, and they have different functions and thereby different target genes. So I think that's something that I'm very much interested to study in the context of oligodendrocytes, which cofactors are activated, and then, as you say, which potentially other transcription factor cooperate with it.
Perfect. I'm really glad to hear that. Yeah, and actually, I see your study you are checking what's going upstream. You find, first you found this that the whole fluid is acting. Then what is in the fluid? SRF. What is acting on SRF? And then you go upstream, and finally this Fgf17, which is very interesting. But now the next question: what is upstream from Fgf17? Do you ask that question?
Yeah, so we touched upon it very briefly in the paper and found that it's secreted by neurons, which was very interesting to us. We thought that it will be secreted by the choroid plexus epithelial cells, which produce the CSF. So now we've uncovered this very complex interaction whereby this youthful factor is expressed by neurons. It somehow, we don't know how it reaches the CSF and then affects the oligodendrocytes, or maybe there's like a gradient and it directly affects oligodendrocytes in young mice. So this whole like triangle between the neurons, CSF, and oligodendrocytes is very interesting, and we still don't know a lot of like the function.
Yeah, I was really surprised and to see that FGF7 is expressed by neurons. And I thought maybe by whatever positive thinking, you can produce this FGF7.
Hopefully.
And become
yeah, who knows? Or or all these aging intervention, right? Like exercise or diet, could it be affecting FGF17 or any other mechanisms? There are thousands of proteins in the CSF that I'm sure are doing very beneficial things in the brain to other cell types or to oligodendrocytes. So I think there's still a lot to uncover.
All right. Yes. So I think, there will be more questions. I hope in the offline we will publish this.
Yeah, of course. So so don't hesitate to contact me by email. It's stated here, and I would love to discuss.
Finally, maybe just a little final, comments. When we are going to have some therapies which will rejuvenate our memory.
Yeah, so specifically for this study, I think it's still relatively far from clinical studies. We still need to understand, as you said, where is FGF17 produced? How can we best manipulate it? What are the doses? And of course, check more adverse effect, as was mentioned here in the Q&A. So we still need to do more basic biology studies to understand the mechanisms before we move on to the clinic with this. But I'm very optimistic that one of these leading rejuvenation strategies that seem to be very effective in model animals will be translatable to humans.
To the human. Yeah, let's hope and wait for that. Thank you very much. And I'm forwarding back to Edgar.
Yeah, thank you very much for this exciting talk again and for the interesting discussion. Thank you very much for joining us today. We just took this as the opportunity to check in Google Scholar and found that there were altogether about 20,200 articles published that made use of the Transfact database. Of course, today we were learning about a very particular and exceptional example. Thanks again for that. And I would also like to announce, making use of this opportunity, that we are going to offer a 1-week free access to the Transfact database to the attendees of today's seminar. You will be contacted by email on that. So thank you, Tal, very much again, and all the best, my best wishes for your further work and your career and your future and everything. Thanks a lot.
Thank you very much again for the invitation and for maintaining the Transfact database. It was very helpful.
Thank you so much. See you at another opportunity. Thank you. Thank you all.
Bye-bye.
Bye.
Goodbye. Bye.